Christie. I'm a graduate student with Sharon Strauss at UC Davis. I feel like to some extent I'm preaching to the choir. We're all interested in speciation, so I'm going to sort of try to blast through this introduction and spend a little more time with the data. But I know that we're all fascinated by this diversity around us, and one big question is, where does this diversity come from? So what do we know about speciation, the process by which new species originate? If we want to understand speciation, we first need to define species. So the biological species concept is helpful here. It's defining species as reproductively isolated entities, and this gives us a clear research program. So if we look along this branch, ancestral lineage, we come to this node or this splitting event that's characterized by the evolution of reproductive isolation. So to understand speciation, we need to understand something about this evolution of reproductive isolation. Okay. And then I think one other interesting thing that I'm particularly fascinated with is we know there's lots of potential barriers to gene flow along the organism's life history. So the early acting prezygotic barriers that are extrinsic, that may be affected by the environmental or ecological context that species or populations are in. And then we have these intrinsic, these post-zygotic barriers. So in my research, I've been very interested in sort of comparing and contrasting and trying to understand the interplay between these two things. So here, thanks to David Lowery, I'm showing essentially a cartoon of the speciation continuum. So on the far left side, we have interfertile populations, and as we move to the right, reproductive isolation is continuing to evolve, and eventually we result in these completely independent lineages. So lineages that are intrinsically isolated, that can no longer cross. I think the important thing to remember here is that, at least in many cases, speciation is gradual. This is a process. And I think that's sort of led to a difficulty in studying speciation. So we're trying to infer something about a process that's taken time by looking at single points along that continuum. So we may be studying divergent populations here. We may be studying a species pair here. It's hard to make emphasis about this whole process by looking at single time slices. So one, I think, elegant method to address these difficulties is these comparative analyses of reproductive isolation. So in these analyses, we have multiple species pairs within a clade, and we're quantifying reproductive isolation between many different species pairs. So essentially, we're taking lots of time slices across this continuum and trying to paint a picture of how reproductive isolation evolves over time. These approaches have been quite popular in animals, sort of pioneered by the work of Coyne and Orr. There are a few earlier examples, but that's sort of what started the industry. Just to orient you to these graphs, because we will be seeing a number of them. On the x-axis, we have genetic distance. So this is a proxy for divergence time. So values of 0, there's no differentiation, no genetic divergence. And as we move along this x-axis, these taxon pairs are older and older. And on the y-axis, we have some metric of reproductive isolation. So a value of 0 would be no reproductive isolation. These things are fully interfertile. And a value of 1, these things are totally reproductively isolated. Okay, so a common finding in almost all of the animal literature is this positive correlation between genetic distance and post-zygotic intrinsic reproductive isolation. There have been fewer studies in plants, but as we just saw, they're becoming more and more common. And findings are a little mixed. So sometimes here we have some sexually deceptive orchids. We have this nice linear relationship, or positive relationship anyway, that we often see in animals. And here in another group of food-deceptive orchids, we don't see that relationship. And one of the interesting things to me is, well, maybe this is indicative of the underlying genetic architecture. So we know that plants, a lot of strange things can happen. We can have polyploidy, we can have chromosomal rearrangements, we can have mutations of large effect, and we can also have genic incompatibility. But one potential explanation for this lack of a correlation is due to some different underlying genetic architecture in plants than in animals. And then I just showed one more image here from, to my knowledge, the first comparative analysis of reproductive isolation in plants from Leon and Moyo. And the point I wanted to make here is that many of these studies have also been restricted to, maybe a single or maybe a few barriers to gene flow. So in this case, we're looking at post-zygotic isolation. We're only using basically pollen viability. So my goal was to apply this method thoroughly to a plant clade and to really look at many multiple barriers that act on an organism's life history. I'm doing this in California Jewelflowers, Streptanthus. This is a really cool group of roughly 35 to 40 species. They're largely annuals. They're largely endemic to California. They occur in harsh habitats, arid habitats, and interestingly, we have more serpentine endemics in this clade than any other genus in California. Importantly to this study, we have a well-resolved and well-sampled phylogeny, thanks to Iglu Kachu, and that really lets us know who's related to who and how divergent they are. So, how is intrinsic reproductive isolation related to genetic distance in this plant clade? That's my first major question. And then secondly, what are the relative strengths of ecological barriers versus genetic barriers? So we know that potentially a taxon parent plants, I'm sorry about that, may be largely reproductively isolated because they're adapted to different climates. They may have different flowering times, different pollinators. There may be some pollen pistil interactions. And then we have these later acting, these post-zygotic barriers. Perhaps there's intrinsic incompatibility at a variety of stages. Can you even set viable seeds? And how well do those viable seeds do? And sort of can they persist? So really briefly, essentially for many species pairs across the clade, I'll talk a little more specifically in a second about which pairs, I quantified these pre-pollination barriers. So I was able to quantify essentially climatic niche overlap by building species distribution models. I was able to calculate flowering time overlap just by looking at publicly available flowering time data. And then I sort of created this PCA of floral morphology, which I hoped was going to act as a proxy for pollinator differences. So how different are these flowers physically? Maybe that would tell us a little bit about the difference in pollinators. And then for these later acting barriers, so post-pollination barriers, these things I measured directly in the greenhouse. So I made a bunch of hybrid crosses. For each of those crosses, I was able to say, okay, how often did fertilization occur? If fertilization occurred, were there any viable seeds? How many viable seeds were there? So I collected all those seeds, and I grew up the F1 individuals the next year, and I said, okay, do those seeds germinate? Are they viable? Do they survive to flowering? And if so, is the pollen fertile? So there I just show an image of some viable pollen and some inviable pollen in an F1 hybrid. Just very briefly, I used these species in the crossing experiment. They represent about half of the clade. We collected, the Strauss lab collected wild populations from throughout California. I grew these up in the greenhouse. And I won't go into the nitty-gritty, but I sort of made 3 sets of crosses. I made clade-wide crosses, so relatively distantly related organisms, indicated by the red box. I made crosses within a single subclade, and then I also made crosses between sister species. So I'm trying to span all of the genetic distance within this group, in this case on the order of 5 million years. I'm going to skip that slide. So in this hybridization experiment, I made reciprocal crosses, so each parent acting as both the mom and the dad, and that encompassed 39 distinct interspecific species pairs as well as the within-species controls. And what this histogram shows you, so these closely related species are essentially sisters. These species here are essentially that subclade in which I crossed each of the individual members, and these are essentially crosses that span broad genetic distances within the clade. So to answer my first main question here, I'm plotting intrinsic post-zygotic reproductive isolation as a function of genetic distance. So remember here, very closely related, and a value of 0 would be no reproductive isolation. And I just wanted to note, I'm plotting the raw data. So each point here represents a species pair. I think it's more intuitive to think about it this way, but I've analyzed this in a phylogenetically corrected context. So all the correlation coefficients and p-values are represented in the phylogenetic independent contrast. But in any case, here we see the strong positive correlation between genetic distance, between reproductive isolation and genetic distance. So this is consistent with the pattern we've seen in most animals and also some plants. Okay, so next I did the same thing for these prezygotic barriers. So here for climatic niche, we essentially see no relationship between genetic distance or divergence time and reproductive isolation that's resulting in different climatic niches. Similarly, we see no relationship between phenological overlap and genetic distance. And this point will come up in a second, but I just wanted to bring your attention to this point. So here we have a very closely related species pair that essentially flowers at exactly the same time. Yet here we have another very closely related species pair that has totally non-overlapping flowering time. We'll come back to that point in a second. In this group, we have sort of very, very strong floral conservatism. So we actually can predict the difference in floral morphology. Whether or not that's a good proxy for pollinator differences, we can talk about that. But we see essentially no young species that have these big shifts in floral morphology. That might be consistent with pollinators. And then fertilization successes, sort of a proxy for pollen-pistil interactions, were sort of marginally significant. The jury's still out. Okay, now I wanted to move on to the post-zygotic barriers. So in this first panel, we're just looking at viable seed set, period. Did the cross result in any viable seeds? And I've basically coded significant values in the blue. So again, we're seeing this positive correlation between reproductive isolation, just does the cross work, and genetic distance. Here I'm showing the same thing for seed mass. How many or how much viable seed mass did the cross produce? Interestingly here, so then I planted what appeared to be a viable, fully formed seed. And interestingly, regardless of how distantly related those two parents were, it looked like a good seed. It was a good seed. So here we have some really distantly related parents that the seeds are germinating just fine. So when does that sort of hybrid problem happen? Well, that happens a little later in the life history. So then when I grew those F1 seeds out, again, we sort of get this strong relationship. So very distantly related parents, maybe they can make a seed, but then that individual F1 rarely survives to flower. We can sort of track this through the life history. Here, we have a very distantly related parents that made a good seed that actually made flowers. We're losing a little power here. Very few crosses survive to flowering and basically make any viable pollen. So again, these distantly related parents, if the hybrid makes it that far, often the hybrid. Okay, one last minute. Then I essentially want to compare the strength of prezygotic barriers versus postzygotic barriers. And the take-home point here, since I'm running out of time, is while these prezygotic barriers may not evolve predictably, they can still be very strong in young species pairs. So we have these barriers, these early acting barriers are equally as strong as these intrinsic barriers. I'm going to just jump all the way to the punchline, but I think earlier we have this tension between postzygotic barriers evolving in a predictable fashion as a function of genetic distance, and prezygotic barriers evolving idiosyncratically. So depending upon the context of your species pairs, I think the real cool stories may be in the interplay of these two kind of types of barriers. So I know we're almost out of time, but I'll just thank you for your time and open up. Talk to me later.